Hey everyone, I want to spend a few minutes going through IB MYP science subjects, criteria, and talk a little bit about the scientific method. So, within the IB sciences, we have our four criteria, our four rubrics that we measure science thinking skills on. Criterion A, known as knowing and understanding, criterion B, inquiring and designing, C, processing and evaluating, and finally, criterion D, reflecting on the impacts of science. If we were to put these in other terms, right, we know that with criterion A, there's some basic foundational scientific information that we need to know in order to make sense of reading we do, videos we watch, observations we make, experiments we do, things like that. Criteria B and C together are sort of the heart of scientific thinking, right? Inquiring and designing, meaning how do you think through the process of setting up an experiment and then design that experiment from start to finish? Goes along with criterion C, as we finish conducting an experiment, how do we collect the data and make sense of what the data are telling us? Right? And once we do that, then we have some results to determine how it may affect people, the environment, etc. And that is where we measure with criterion D. So most often, scientific thinking is done through the scientific method, right? And there are a variety of different diagrams and charts and processes that show scientific thinking. But in general, right, we follow sort of a process of noticing something in the world around us, asking a question about it, doing a little research in order to understand what's involved, and then thinking about if I were to test this somehow scientifically in an experiment, what might I find out? And what do I think will happen before I begin? What is my hypothesis at the beginning? Then we conduct our experiment, and this is where you see the arrows start to go in different directions and repeat a little rather than just follow a straight linear flow. If something is working well in our experiment, that'll lead us to data that we can collect and analyze. Sometimes, though, we need to think about how to redesign the experiment, maybe keep something constant that we hadn't thought of in the beginning, or add more trials to make sure our data are reliable. Right. And once we have the experimental procedure and the setup uh, thorough and accurate and complete, then we can move through analyzing the results and drawing conclusions. So, what I'm talking about here is that science is a process or a way of thinking, not just facts about living things and the environment and energy and so on, right? Scientific thinking tries to get at the heart of a cause and effect relationship, understanding what things happen and why they happen and how they happen, right? And we do that through thinking about variables and which things are having an effect on what we're observing. So, in this first part of our chemical reactions unit, we're going to talk about setting up an experiment. You all will conduct one to demonstrate your understanding of chemistry and the scientific method. So, I want to go through a few of the basics of that, what that means and might look like in your work. And as I've talked about collecting data in an experiment, there are two main types of data or observations or measurements that we can be aware of: quantitative and qualitative, right? And both are valuable. Quantitative measurements or observations or data involve those that we can measure with a number, a temperature, or a quantity or volume, something like that, time. Qualitative observations, on the other hand, are those that we observe but may not have numbers, hard numbers with them. We might observe bubbling in a chemical reaction or a color change. We may use our senses like touch to see whether things are warm or cold, which leads us then to setting up a good testable question, right? Not all questions can be tested through an experiment, right? It'll need to have some sort of qualitative or quantitative data we can collect. There will need to be the ability to manipulate or change things to see if they are having an effect, a cause effect relationship on the outcomes. And then we need to ensure that the question has enough explanation to it so it's clear as to what we're testing. So here's a common frame for how we might set up this question. What effect does something have on something else when we do it in this way, right? So what effect does iPad or phone screen brightness have on the length of time a teenager can focus on what's going on that screen when we make sure that they're in the same type of environment and learning the same thing, right? So that's pretty clear as to what we're going to test, and starts to explain some of the things we know we will have to be aware of and keep constant or the same. So, because we've made observations and we did a little research, and we kind of are wondering what's at the bottom, what's at the heart of this question, we're going to set up a guess or a hypothesis before we begin, right? And the hypothesis is it's based on some things you've learned in the past, right? And it should sort of describe what results you think will happen. It's a statement, not a question, right? We've already set up a testable question. So I might format my hypothesis something like this. If screen brightness is set above 50%, then students will be able to focus longer because they will see things more clearly and not get distracted as easily. All right, that could be my hypothesis for this example experiment I'm talking about. Now, next, what I'm going to need to do is identify different variables in the experiment. And the power of variables in a scientific experiment is that we can try to figure out as many variables as possible and keep as many of them the same as possible, so we can test just one at a time to see if it's changing what happens. So the variable that we are going to be in control of and change to see if it's making an effect is called the independent variable. Right, so in my screen brightness experiment, we can change the screen brightness. The thing that's going to happen one way or another, and we don't exactly know yet, is how long the students will pay attention. Right, so if I set the brightness at 50%, I can write that down and then time how long. If I set it at 90%, right, you get the idea. The variable that we're going to measure as a result of that will be the time students are paying attention. Okay, and that'll change depending on how bright we make the screen or some other factor that maybe we haven't identified yet. This variable is called the dependent variable. It depends on the thing you change. All right, then the last thing that we need to be aware of in setting up a really good experiment is that there are parts of the experiment we will want to make sure we keep the same, right? Those are called constants. Keep them constant every time you run your experiment, because if I'm trying to figure out if screen brightness has an effect, I want to make sure that things like caffeine and sugar and number of hours of sleep and room they're in and teacher they're learning from, all of those things are not causing the effect. I really want to isolate things just down to, is it the brightness? Okay, so I'm going to try as best I can to keep as many things possible constant, so I can be sure that those are not causing the results. All right, when you run an experiment just one time, you can't always be sure if it was a consistent result or if it was just sort of a random result. So number of trials or times that you repeat an experiment can help increase the reliability of what you're figuring out. And often we will have a control trial or something that is kind of kept in the normal version, a baseline, so that we can compare all of our other results to it and understand how different the results are that we might be measuring. So some common examples here would be if we're testing different amounts of fertilizers on plants or kinds of fertilizers, we want to give one plant just plain water to see how much it'll grow with just water, rather than mixing the fertilizers into different kinds of water and that being the effect. Or often when they're testing new medicines, they will use either no medicine or a placebo or a fake type of medicine to see if it's the medicine causing the effect or not. These are known as control trials. All right, so that's sort of the first half of the scientific method, and we'll pick this up in another video to go through the other parts.